Good evening, and thank you so much for this kind introduction, Volkan. Let me begin with expressing our gratitude for the warm welcome and hospitality that Annette and I have received from everyone here at the Kelsey Museum over the past few weeks, and especially our dear friend Laura, who hosted us in her lovely home. We've been visiting Ann Arbor at the Kelsey Museum for various projects since 2016, and coming back here in a way always feels like coming home. Therefore, I'm immensely gratified to be here tonight to talk with you about our very exciting joint project, AGROS. So, what are we going to do tonight? Well, my objective is to give you a bird's-eye overview of the AGROS project, of which the acronym stands for Agriculture, Diet and Nutrition in Greco-Roman Egypt, Reassessing Ancient Sustenance, Food Processing and Malnutrition. I would like to share with you what this large and intricate project is about and who is involved doing what. I will furthermore discuss the project's rationale and historical context and explain how it relates to its predecessor, Rethinking Roman Nutrition. Of course, I will also share with you some results, insights and challenges from both projects. And I will do all of this from my role as the coordinator of the AGROS project. The AGROS project consists of 6 teams from 5 different universities. The overall project is highly interdisciplinary and includes archaeobotanists, archaeozoologists, general archaeologists, historians, Egyptologists, but also food chemists, food bioengineers, veterinary scientists, and microbiologists. And our core team consists of about 20 people. The project itself consists of 2 main topical pillars, and each team has work packages in both pillars so that there really is an integrated interdisciplinary approach. That has its challenges, as different disciplines speak very different languages, but in my opinion it is absolutely worthwhile, as it also makes the project truly interdisciplinary. Pillar 1 of the AGROS project focuses on holistically reconstructing vital elements of the agricultural and food economy of Karanis within its Fayumi and Egyptian contexts. And besides looking at the textual sources, this includes the assessment of the botanical and zoological collections from Karanis here at the Kelsey, as well as the assessment of the excavation records kept here to better understand chronology and context. It also includes nutritional and anti-nutritional analyses on the ancient plant remains from the collection. And why we do that I will explain in a little bit. Pillar 2 then focuses on reconstructing ancient food processing and preparation practices and, where possible, recipes, and then replicating those in the biotechnology lab. Afterwards, we assess the nutritional and anti-nutritional profiles of the foodstuffs that were consumed to better model ancient diet and nutrition. But we also look for elements that can be used to improve the nutritional outcomes of modern industrial foods. As I mentioned, AGROS is the much larger successor of the previous Rethinking Roman Nutrition project. That project was our first joint foray into historical diet and nutrition. As we were conducting the project, we realized there was much more complexity, but also much more potential than we had originally imagined. In that sense, the interruptions amidst the project caused by COVID were not only a delay, but also a blessing in disguise, as they gave us the time to take a step back and think about things. And what we realized was that it would be opportune to expand upon the project, and after we won AGROS, we could. The projects are by design dovetailed together, so we will discuss some elements and results of both in this talk. I will, however, first begin with outlining the rationale and questions behind the projects and provide some context from the historical debates they engage with. As those of you with a historical background in ancient history will know, over the past few decades, there has been a major shift in the debate on the performance of the Roman economy. Previously, primitivist scholars such as Moses Finley had rather grim ideas about the ancient economy and viewed it as stagnant, underdeveloped, and underperforming, and with living standards to match. But this perspective began to give way under the influence of mounting archaeological evidence suggesting the contrary. And today, most Roman historians believe that the Romans experienced at least a modicum of economic growth. What persisted, however, was the accompanying negative view of Roman diets as being unwholesome and deficient in key nutrients. These deficiencies were attributed to a dietary reliance on staple crops such as cereals and pulses that were held to be intrinsically poor in nutrients, especially in terms of minerals such as iron and zinc, copper, magnesium, and others. At the same time, they were seen as being rich in anti-nutrients such as phytate that reduced the bioavailability of the nutrients that were present. Ancient historian Peter Garnsey is one of the main proponents of this view, which he aptly discussed in his seminal Food and Society in Classical Antiquity. This Garnsian model has had a lasting influence not only in ancient history, but also in the archaeological sciences, including osteology and human stable isotope studies, to explain cases of malnutrition or food-related diseases. And while Garnsey did very important work and his model has its merits, I didn't agree with it on grounds of it being possibly anachronistic. What is very important to emphasize here is that we're not arguing that there was no malnutrition in the ancient world, because it is well attested archaeologically that malnutrition occurred and was widespread. It would also be highly unusual if it didn't, seeing the prevalence of micronutrient deficiencies around the world today, as you can see from some of the examples listed on the slide. It would seem that a degree of malnutrition, sadly, still is part of the human condition. Our argument is that the causes for ancient and modern malnutrition are categorically different, and that the eagerness with which the current historical paradigm seeks to scapegoat cereals and pulses may be unfounded. So what we wanted to know was if this negative appraisal of the nutritional qualities of ancient cereals and pulses was justified. This raised the question as to what sort of evidence do we have and what do we really know about the nutritional properties of ancient crops. The answer was surprisingly little, and certainly much less than we should like. The problem is that the data underlying the pessimistic model of ancient diet and nutrition is entirely based on modern crop nutritional and dietary studies stemming from the second half of the 20th century. Applying such data to the ancient world is problematic for several reasons, but especially due to the Green Revolution of the 20th century and the major changes our crops underwent during this time. Throughout history, the amount of available nitrogen as a plant nutrient was the limiting factor on crop productivity. The amount of available nitrogen depended on the amount of animal manure available, putting a cap on output. This all changed with the invention of the Haber-Bosch process, with which cheap artificial nitrogen fertilizers could be produced. The new limiting factor on output, at least for cereals, then became lodging. When cereals receive larger amounts of nitrogen, their ears become heavier, making the plant top-heavy and prone to falling over, leading to a loss of yield. As most cereals were long-stemmed in the past, this was a serious problem. This made developing cereal cultivars that were shorter and therefore less prone to lodging an important objective, even with strategic implications to the world powers at the time. Japanese researchers in the 1930s had much success in this direction, developing semi-dwarf short-stemmed cultivars, most notably the Norin 10 cultivar. After World War II and the American occupation of Japan, this research was confiscated by General MacArthur and brought to the United States, where a promising young researcher named Norman Borlaug and his team continued this work. Excuse me. So one of the main consequences of these efforts in the long run were that we went from cereals that were tall, like on the left, to cereals that were small on the right. Besides increasing yield dramatically, Borlaug and his team also solved a myriad of other issues in cereal cultivation. This series of advances in agronomy we refer to as the Green Revolution. Though not the most well-known, it represents one of the most technologically important advances of the 20th century, and in agriculture perhaps the most important one since the Neolithic Revolution itself. It helped prevent major famines in the later 20th century and paved the way for the post-World War II demographic explosion, earning Borlaug the title of the man who saved 2 billion lives, and a Nobel Prize to boot. As you can see in the graphs behind me, these advances dramatically increased wheat yields worldwide, with Borlaug and his team, for instance, famously more than doubling India and Pakistan's yields between 1965 and 1970, and many other crops would soon follow suit. It was realized only much later, however, that these short-stemmed, high-yield varieties, nutritionally, had some unexpected and unintended drawbacks. Cereal chemists discovered that the micronutrient content of these varieties was much lower than that of their pre-Green Revolution counterparts, especially for important minerals such as iron and zinc, calcium, magnesium, and copper. The difference became greater as they analyzed specimens from further back in time, using 19th century herbarium specimens. As yields went up, mineral content went down. Part of this difference could be explained from a dilution effect. As yields increased, the amount of trace element available per plant or per weight of product decreased. More importantly, it was also found that short-stemmed varieties were less efficient at routing minerals to their grain kernels, negatively affecting the nutritional profile of the consumed grains. So increase in productivity came at a price. For some minerals, the reduced micronutrient content in recent history sprang from other causes. The selenium content of cereals, for instance, is greatly influenced by sulfur dioxide pollution that interferes with the uptake of selenium by the plant. Ancient cereals and pulses were not subjected to the same crop manipulations and other changes as modern crops were during the Green Revolution, nor were they subjected to the types of pollution brought on by industrial society. These phenomena and their effects were specific to the 19th and 20th centuries and cannot be transposed onto the ancient world, as this would be anachronistic. Today's cereals and pulses that contain up to 30% less of important micronutrients than their 19th century counterparts can therefore not serve as a valid basis for modeling the nutritional profiles of ancient crops. We can, however, also not just apply the 19th century data to our models instead. That could be equally anachronistic, as there are over 1,500 generations between the Roman and 19th century crops. As a result, we are presented with the predicament that we do not have any reliable data on the nutritional properties of ancient crops. So how could we solve this predicament? Well, by trying to obtain empirical data from ancient crops. That, however, was more easily said than done. The problem is not that we don't have any ancient plant remains, but that these typically are preserved in a charred and carbonized state that is not necessarily representative of its chemical composition before charring. If we wanted to measure nutrient content, desiccated, so naturally dried-in remains, like those one could find in hyper-arid environments, would be our best bet. Such materials, however, and especially collections large enough to allow for the destructive analysis of even small numbers of specimens, are incredibly rare. Fortuitously, such a unique collection exists for the Greco-Roman village of Karanis in the Fayum Depression in Egypt, that is held here at the Kelsey Museum. As most of you who are based here at the Kelsey will know, excuse me, Karanis was a village that was built on the edge of the desert, as in Egypt people do not build houses on land that can be irrigated. Under these hyper-arid conditions, the preservation of organic materials is superb, making Karanis a veritable Egyptian Pompeii. Besides copious amounts of papyri and objects from everyday life, numerous sealed storage bins and other vessels containing supplies of seeds were preserved. During their excavations in the 1920s, a time during which the archaeologists were typically not interested in such materials and discarded them, the Michigan team carefully excavated them and have preserved them in pristine conditions here at the Kelsey Museum ever since. This is truly a one-of-a-kind collection, all the more since comparable sites have since been destroyed without having been similarly sampled. And from the pictures in the back, you get an impression about the amazing level of preservation of this material. So with this, the stage was set for commencing our first project, Rethinking Roman Nutrition. Within this project, we had three objectives. Of course, we wanted to assess the mineral content of the crops in comparison to their modern counterparts. But it was very important to first get the chronology and dating of the samples right. That was all based on relative dating up to then, and it was known that there were some issues with that. Moreover, we wanted to also look at trends in the data within antiquity, and not just compare ancient and modern data. Besides looking at nutrition, we also wanted to look at agricultural practices at the site through stable carbon and nitrogen isotope analysis of the remains, but I'll get to that in a bit. So we did analysis on what I will refer to as the first batch of subsamples we took from the collection. We back then focused first on samples that were obviously large, huge, large boxes with tens or hundreds of thousands of specimens. There was no time within that project to look at the botanical collection that had never been studied in detail before, as that's a very large task in its own right, and one that we're now conducting within the AGROS Project. And there's of course the ethical issue of destructive analyses. So over time we at first took samples from the larger contexts, and then COVID hit. Last April and May, when in the context of AGROS, we, and with we I mean especially Laura and Annette, were back to work on the botanical collection, we found that a lot of these small samples were not that small. In these unsightly tiny bags with sand and seeds, there were sometimes a few thousand grain kernels. That was really a game changer, because then it became justifiable to select, say, 10 specimens out of 2,000 or 3,000 to do all of these analyses. So we could do a bunch more samples than we initially thought, which is better for range, representativity, and statistics. Oops. This work started over the summer, and those results should be in soon. And we're really excited to see if and how this changes or nuances some of the things that we've seen thus far. But that's why today we still reflect only on the trends and observations from the first sample batch, and we'll be back with more, hopefully very soon. So, radiocarbon dating of the samples from the Karanis Botanical Collection showed some unexpected and spectacular results that were published just before the summer in a paper spearheaded by Laura. This showed that the archaeobotanical remains in the collection are from a later date than expected. Instead of hailing from the 1st and 2nd centuries AD, they mostly originate from the 4th through 7th centuries AD. This has great consequences for the chronology of the abandonment of Karanis, which was always thought to be a few centuries earlier, and may reframe the debate about the late antique decline of the Fayum. So, very exciting stuff, and more dates are on the way. For the minerals, we used inductively coupled plasma spectroscopy, or ICP-MS, to measure the content of nutritionally important elements such as iron, zinc, and others, after having first optimized the methods on modern materials, so as to not waste precious ancient material. In fact, to maximize the data output from these samples, we more or less measured everything we could at the same time from the same material. So also elements that are not immediately nutritionally relevant, but may give environmental or other data, were also measured as we could do that at the same time anyway. So we did this for a number of different crop species, so that together with the dates, we can not only look at changes over time, but also between crops. And all these results we will compare and are comparing with modern data from the literature and analyses of modern specimens that I had previously collected on the Fayum, and often from fields very close to Karanis, like the field you see here in the background, this grain field. So the most important observation from the samples thus far is that the ancient samples, on average, have a far greater mineral content than their mother counterparts with respect to most micronutrients. The table on the right shows the average difference in percentage for some of the nutrients across the taxa measured, just to give an impression. This shows, for instance, that for iron, the average content across samples and taxa measured is 45% higher than that of modern cereals and pulses. A point to take away here is that this substantial difference even extends well beyond the upper level of a 30% difference observed between the samples from the 19th century and the post-Green Revolution samples. Especially for important nutrients such as iron and zinc, this difference is really significant and matters. Additionally, it would seem that the ancient samples have far smaller amounts of certain metals and metalloids than their modern counterparts. And these differences can likely be attributed to modern pollution, likely coming from industrial waste, pesticides, etc. Very interesting is that the amount of natrium or salt in the ancient samples is far greater than in the modern ones, on average by as much as 93%. Modern agronomic studies suggest that high natrium content in cereals is particularly related to soil salinity. And salinization is one of these factors of interest in the discussion on the late antique or alleged late antique decline of the Fayum. With stable isotopes, we move away from nutrition for a moment. As plant-stable nitrogen and carbon isotope analyses are respectively used by archaeologists to reconstruct manuring practices and plant water status. Therefore, these tell us more about agricultural practices and environmental conditions. Isotopes are variants of an element, and the stable ones occur in nature in fixed ratios. But all sorts of processes can alter these ratios, which we call fractionation. And as archaeologists, we're especially interested in changes that are caused by human activities, or say something about human environments. We can measure isotope ratios with a mass spectrometer and express differences from the natural ratio in parts per thousand, or per mille, as we say, using the so-called delta notation. And we talk about these samples as being depleted or enriched in something. And again, we could compare our ancient results with modern reference material, including those collected at the fields close to Karanis. So, very briefly, carbon isotope ratios can be used to assess plant water status and can be helpful in identifying water stress in particular. This all has to do with the exchange of gases during photosynthesis, so basically the breathing process of the plant. Normally, the heavier 13C isotope is discriminated against during this process, as the plant preferentially assimilates the lighter 12C and breathes out the 13C. If the plant is, however, faced with drought, it closes its stomata, which are like little mouths on the leaves. And it closes them more frequently and longer, so as to not lose too much water. The 13C will then also be used up, and the ratio in the plant tissue, including the seeds that we find, will change. Well then, what do these results thus far say about plant water status at Karanis? Well, most plants actually appear to have been relatively well watered, and there is no apparent evidence for significant water stress. Though there is a difference with the modern crops, which are typically even better, even more well watered, thanks to pumps and fossil fuel irrigation techniques as opposed to the more manual ones that were more popular back then. Again, this is very interesting in the context of the alleged late antique decline of Karanis and the Fayum, in which water problems due to climate change or water resource overexploitation take a key position in the argument. And at least in these samples, that doesn't appear to be a major issue. On the other hand, we may be dealing with a lack of negative evidence. In years with very bad droughts and very poor harvests, there's less grain to go around, and most of it will be used up for food or for sowing seed, and we have a smaller chance of finding it. From years with bumper crops and bumper harvests, there's much more material and more stuff that can be stored for a rainy day, or rather a not so rainy season. So we're more likely to find it. The additional results in assessing the correlation with salt content may help us move forward here. The nitrogen isotope ratios can help us recognize the agricultural practice of manuring, and more importantly, its intensity, because that people were doing it, we know. The process here is that as manure is spread out on the field, some of the nitrogen will volatilize, so meaning evaporate, in the form of ammonia. And during that process, the lighter 14N isotope more readily evaporates than the heavier 15N isotope. Therefore, the soil gets enriched in 15N compared to the naturally occurring ratio. And so do the plants that grow on that soil and that use the nitrogen to grow. At least that is how it works with most plants. Pulses, which are leguminous plants from the Fabaceae family, cheat, as they get most of their nitrogen from symbiotic bacteria that live on their roots. They can take up nitrogen from manure, but typically stay close to the natural level, so about that close to the zero difference rate. So if we look at the nitrogen isotope results from Karanis samples thus far, what stands out really is that the enrichment of some of the samples is really high. For cereals, we frequently see 10 to 15 per mil enrichment, and that is quite extreme. As a comparison, modern manured local samples are around 4 per mil. The ancient pulses, with values between 0 and 3 per mil enrichment, are less extreme, but still higher than expected and more than the modern ones that gravitate around 0. So how do we explain this? The traditional archaeological isotope logic would be to say higher nitrogen enrichment means more manuring, so higher yields and very intensive productive farming. That could well be, but this enrichment is so high that there may also be other things at play. Are people from Karanis, for instance, using manure that is already very much enriched to begin with, such as human waste? Or are we dealing with something called trophic upcycling, where enriched manure leads to enriched crops that are consumed by humans and animals, which leads to more enriched manure, and so on? Especially if you use a relatively small area of irrigable land continuously over centuries, that could be something that might be at play. And lastly, but then we get a bit more technical, nitrogen enrichment itself is not a direct proxy for manuring, but for ammonia volatilization. There is modern agronomic research that suggests that windy or warm and dry conditions increase this volatilization, and so does inefficient collection and storing of manure. And that's exactly what you as a farmer don't want to happen. You want your nitrogen in the soil where the plant can access it, not out in the atmosphere. So in that model, high enrichment can also be a bad thing, an inefficient thing. And the confounding thing is, of course, that these different factors are not necessarily mutually exclusive, so more of them might be happening at the same time. We will be moving forward on these questions, and the new results from the small samples will be extremely valuable in this respect, also because they include a dung sample. And also work that's now being carried out in the context of AGROS will contribute to our interpretation, such as animal isotope analysis and the protein content and composition determinations of plant remains, as protein is ultimately nitrogen and in plants relates to manuring. Lastly, the papyrological work that is being conducted on manuring practices will hopefully provide some insights as well. And that makes for a very nice transition into the AGROS project proper. Excuse me, I have a very itchy throat, so I keep on drinking water. So as I mentioned, when we were working on the first project, we realized that there was not only a lot of potential in doing more work, but it was also essential to do it. The first reason for that is that there are many nutritional and anti-nutritional factors besides minerals that are important to human nutrition. And some of these may even interact. There is, for instance, the anti-nutrient phytate that chelates or binds minerals so that they are not as bioavailable to the human. Ancient grains may have more minerals, but if they would have more phytate as well, you might still be worse off. The second point is that the plant remains constitute the raw ingredients people would use to make their foods. But we humans don't eat most of our foods raw. We process and prepare them and change them chemically in the process. And these historical processes often deviate from our own, so they're hard to model. In fact, they might be more effective in reducing anti-nutrients and generating nutrients, which is why bioengineers are so interested right now in traditional techniques and exploring their efficacy. So if we really want to understand ancient nutrition, we had to take this into consideration as well. Moreover, foodways arise in a wider socioeconomic, environmental, and cultural context. If we wanted to truly understand or model ancient diet and nutrition, we needed an integrated, interdisciplinary, and holistic approach, or in sum, we needed a bigger project. With that in mind, we designed AGROS. And in the first part of Pillar 1, if you remember the pillars from the start of the talk, we make a holistic assessment of the agricultural and food economy of Karanis and the Fayoum within its Egyptian context. And that consists of multiple parts. First, there's the study of the Kelsey excavation records to reconstruct the archaeological context and improve upon the chronology at Karanis. And this work is conducted by Laura and Tyler Johnson. And besides work on the records, this will also include a new series of radiocarbon dates. Then there is also the qualitative and quantitative assessment of the archaeobotanical materials, on which Annette Hansen and Laura Motta are also presently working during this visit. I will not say too much about that here, because on December 6th, Annette will give a flash talk on Zoom about that. So keep an eye out for the posting or advertisement with the link in it. It will be very exciting. From January 2025 onwards, Mauro Risetto will be joining the project, and he will start with an assessment of the faunal remains in the collection. And hopefully we will also be able to sample for animal carbon and nitrogen isotopes. And lastly, the papyrological team from Leuven and the Egyptological team from Namur will be scouring, or are scouring, the papyri and temple texts from the broader period and region for agricultural and food availability information. The second part of Pillar 1 is devoted to the chemical assessment of plant nutritional and anti-nutritional factors of specimens from the Karanis botanical assemblage. Here we will be looking at the macros, so fat, carbohydrate, protein, but also various vitamins, amino acids, gluten, and fatty acids. In terms of anti-nutrients, we, as I mentioned, will look at phytate, but also at tannins and saponins, and some toxins and anti-nutrients that are specific to particular crops, such as ODAP for grass pea and certain alkaloids in the case of lupine. And this work is mostly carried out by the bioengineers in Brussels and the food chemists in Liège. Lastly, there are two other elements under this pillar. The first is an ancient DNA pilot study on the plant remains we are conducting with external partners at the University of Ghent. And secondly, there will be a study on the strontium isotopes of the animal and plant remains that will be spearheaded by Laura. But as both are not directly on food, I will not discuss them further for now. Instead, I would like to talk to you a bit more about the challenges of measuring nutrients and anti-nutrients in the ancient samples. As we have the material and methods to measure these compounds exist, conceptually this may seem easy, but in practice things are more complex, as they always are. The main issue typically is the amount of material needed for destructive analysis. By archaeobotanical standards, the samples in the Karanis collection are huge. Even those that I earlier called small by archaeobotanical standards, really big. That's how it is. And by chemical standards, the sample amounts needed to perform the existing methods to quantitatively assess most types of anti-nutrients or nutrients are small. The problem is the interdisciplinary confusion in tongues here about what's big and what's small. In that respect, with isotopes and minerals and C14, we're lucky, because these really require only small amounts of material. For example, for mineral analysis, we need as little as 500 micrograms per sample, which is a fraction of one seed for some taxa. But for many others, we need much more material. And that makes sense, because these methods were developed for modern agricultural and food science purposes, and amounts then aren't an issue. You can always call with Monsanto to send another bag, and they will do it. But for us in archaeology, the destructive analysis of material is also an ethical consideration. To give an example, I recently, a few weeks ago, spoke with some of the colleagues at the bioengineering lab about assessing the quantity and quality of gluten in the ancient material and how much they would need. And for their standard method, which is done in triplicate, because they're scientists, they said, oh, we only need 55 grams. That sounds like a little, but it's over 1,100 wheat kernels. So that we're not going to do, and they're now back to the drawing board and see if they can do better with a different method and try something else. But these things always feel a bit like a negotiation. I mean, they always think that we try to give them too little, and we think they ask for too much, so they have a little extra, but that's always an exciting part. Sorry. Then there's the issue of deterioration. Not all compounds are stable, so some deteriorate over time, for instance due to Maillard reactions, and fall apart into other compounds. And those in turn may do the same and fall apart in yet other compounds. So you may have to measure some deterioration product and reconstruct the parameter that you are looking for. And this is all new territory, as no one has looked at these sort of things in material this old. There may also be an issue with detection, for instance, because an existing method doesn't work on the ancient material. The team in Liège, for instance, had an issue with phytate, where instead of the traditional and fairly simple colorimetric method that didn't happen to work, they had to develop a new high-performance liquid chromatography, or HLPC method, instead. And then a different type of problem from the natural sciences perspective is that we are often dealing with relatively small numbers of samples or populations per crop or per crop species. Sometimes we only have a handful of different samples or species, or even just one, even though we may have thousands of individual specimens. Archaeologically, that's not so much of an issue. We have what we have, and in this case, that's more than everyone else ever had. But modern agronomic studies may look at a dozen or hundreds of different populations, especially when testing new methods. So we compensate with additional traditional cultivars from seed banks and from our own reference collections. The solution to these challenges is typically new method development. Existing methods can sometimes be miniaturized to acceptable sample sizes, or alternative methods can sometimes be found or developed. Developing and validating new methods is very time-consuming, expensive, and requires a lot of modern reference material. This is, however, essential for the type of material that we work with, and these new or adapted methods also have intrinsic scientific value, and they also represent project outputs. So it's two ways. It's not just that archaeology gets better out of this, but the natural sciences get better out of this as well. So, and where possible, of course, external collaborations can be a cost and material efficient solution. We just found a partner who developed a new method that would allow us to, at the same time, measure vitamin B3 and 6 different types of alkaloids in only a sample of 2 grams. So that's pretty good. Oops, and here you see one of our test fields on a historical estate in the Netherlands, where we produce some of our own reference material, as well as material for the food processing experiments. We have multiple of such fields in different locations, also in Belgium, in different collaborations with partners on various projects. We currently have several new methods in development, and 2 new ones were just completed. One of these was developed by Ahmed Mustafa for ODAP, and the publication on the method and the article looking into ODAP variation and variability in over 300 ethnographic and modern grass-pea cultivars is now underway. The food experiments with this compound were also completed, as I'll mention in a bit. And with the method tested and true, we can now move on to the ancient material. But it almost took 2 years to get there. So this is an intensive process. The other new method is the HPLC method for measuring the phytate developed by Amira Ali that I just mentioned. She is still rounding off the assessment of phytate variation and variability in over 200 different ethnographic and modern cereal and pulse cultivars, which will be important for interpreting the ancient results, as well as rounding off the food experiments. But she already has some preliminary ancient results, and these suggest that the phytate content in ancient samples was comparable to that in the modern ones. And as I explained, that would be exciting, because phytate stands in the way of the uptake of minerals. So if the ancient samples not only have a greater quantity of minerals in them, but similar or lower amounts of phytate, that would mean that they were also effectively more nutritious. So promising and exciting, we have to see what happens next. We then move to pillar 2, where we are working on recreating ancient foodstuffs. On the part 1 of Pillar 2, we are working on the identification and reconstruction of partial recipes, food processing and preparation protocols, and the conditions under which they were executed. In this, we have a particular focus on products that include fermentation. This is not only because many common basic foodstuffs in the past, and also today, such as bread, beer, vinegar, but also various dairy and meat products are fermented, but also because the study of traditionally fermented foods shows that they lead to different nutritional outcomes than their modern counterparts, and sometimes better ones. So we wanted to know what happened if you, instead of traditional, you went to historical recipes and techniques. So again, the broader archaeological evidence from Karanis is being reviewed, for instance, the grinding stones, cereal grinding implements, as well as evidence from the diverse written sources. And this time we also look at ethnographic analogues and where available at previous archaeological experiments. The next step on the part 2 of Pillar 2 is that we actually recreate the foodstuffs and assess them nutritionally at each processing step. This begins with more or less translating the historical and archaeological information into a practical set of protocols and an experimental design that can actually be executed in the lab. Explicit, let alone clear, recipes from the text are extremely rare. So this is not an easy process and requires a lot of puzzling and compiling different types of data. The next step is that the foodstuffs are recreated in the VUB lab and in our pilot food processing plant, where we have a brewery, bakery, and chocolaterie, and a full-time professional brewer and baker and chocolatier on retainer to help our researchers in their experiments. And then, after, if people still get to that point and not, we will assess the chemical changes that happen in these products at each step. So how much anti-nutrients are reduced, for instance, the reduction of phytate during fermentation was achieved, or how much biogenesis of certain nutrients, such as B vitamins, from bacterial action. And this is stuff where the microbiologists, of course, look at, and they moreover look at how these complex substrates affect microbial diversity. So just let me show you 2 examples, one that's completed, one that's ongoing. The first one on the right is an experiment that Victoria de Bru is working on. And she's replicating a medieval Egyptian non-alcoholic beverage called fouka, which means as much as bubbly, as it was a mildly fizzy drink. And that could be sold in the vessels that you see there on the left. And Victoria started with this drink as it's very similar to an ancient beverage served in a similar sort of vessel that is in the so-called Egyptian menu song. And we looked at it, at the recipe and at the data, and discussed the processing steps and the ingredients with the bioengineers, the people from the written sources teams, and our external partner Mena El-Dori from Cairo, who was looking at a number of these vessels. And we realized that we might be dealing with the same thing, but then over an extremely long period tradition, something that changes, the meaning changes, but the process stays more or less the same. So we're now testing this, if this holds up in practice, by comparing the resultant batches of the drink from the different recipes. And you see an example of that over there. And what was very helpful was that there were many different, explicit, very clear recipes for the medieval drink. So that was really helpful. And what's curious is that these drinks, even though they ferment, and they ferment for a longer time, only seem to produce interesting aromatic compounds. And contrary to regular beers and other drinks, they only start producing alcohol on average after 17 days. So maybe we're looking at an alternative new drink for kombucha. Hey, we never know. That could be interesting. Oops. The second example on the left is one of the food processing experiments conducted to test Ahmed's new method for measuring the ODEP, which is the neurotoxin that occurs in grass pea. And one of the preparation methods was boiling these pulses with a small amount of natron, which is a naturally occurring product mainly composed of sodium carbonate that was widely used in ancient Egypt, both for cooking, for instance boiling vegetables, but also for many other things, including glass making. And in some areas in traditional cuisine, sodium carbonate, or its close cousin, sodium bicarbonate or baking powder, is still used when boiling vegetables and pulses. And in the latter case, they do so because that helps reduce the cooking time. Oops. And we were wondering if this practice could also positively affect reducing ODAP content. As because of its nature, we suspected that an alkaline environment, because sodium carbonate is an alkaline, might be helpful in reducing this ODAP. So we tried it out, and it proved to work quite well, because the ODAP content was reduced by a staggering 81%. And this is an outcome that's also societally relevant. ODAP is still causing problems in countries where grass pea, thanks to its drought resistance, is a common crop and an important emergency food. Today, many people, for instance in Ethiopia, where grass pea has always been popular, are affected by ODAP and neurolaterism, the disease that it may cause if consumed as a staple. But with this low-cost, high-impact method that people can easily use at home, and which likely will have a high degree of consumer acceptance, we may make a positive contribution in preventing this disease in the future. And this may also be a step towards making grass pea more popular as a crop and help increase climate resilience and food security for communities in arid regions where they could really use this crop. So lastly, in terms of project synthesis, on the one hand we will combine and integrate the different elements of both research pillars and use this information to nutritionally quantify historical documents and compose nutritional historical food baskets. Whereas on the other hand, we will try to practically apply and integrate effective methods in modern industrial processes to make them better and more nutritious, and in development economic strategies to help local communities. And with that, I would like to thank you for your attention, and I would be happy to answer any questions you may have. Thank you.